MMS is really proud that we give out graduate scholarships to people in the University of Minnesota and University of Wisconsin systems. We've given out 4 scholarships this year. 2 of the speakers are with us tonight. I also just wanted to mention that this scholarship was originally created in memory of James Swanson and Doris Johannes. So many years ago, they were in our club, and we wanted to do something to memorialize them when they passed. So, all right, our first speaker tonight is Michael Malone. He received his master's degree from the University of Wisconsin-La Crosse. And I'm going to butcher a couple mushroom names here, where he studied Polynesian Xylariales. Previously, he studied, okay, Michael, you're going to have to help me with this.
Oh, Laboulbenian.
Laboulbenian, great, at Syracuse University during his undergrad, and he'll be studying novel fungi of the New Jersey pine barrens at Rutgers this fall. In general, he likes to study the ecology and phylogenetics of obscure fungi, particularly within the Ascomycota. So this presentation consists of 2 research projects that he conducted at University of Wisconsin-La Crosse. And please put your questions for Michael in the chat, and there will be time after his presentation to answer them. All right, take it away, Michael. Thank you.
Oh, thank you for the introduction, and thank you everyone for having me here today to present. And I'm grateful for the Mycological Society to award me this scholarship. So thank you all very much. All right, so this is my master's thesis project. I was working under Professor Dr. Todd Osmundson. He was my mentor for this project. He's been an amazing mentor, and he studied Polynesian fungi, specifically like French Polynesian fungi in the past. And I wanted to kind of do an offshoot of a previous biodiversity project that he participated in, and I was also interested in looking into a taxonomic group of fungi that would just kind of be like the most like oddball group of the specimens that he collected. So I chose the Xylariales. So this is my thesis project that was the research effort towards my master's degree, the phylogeography of Polynesian Xylariales. So you've probably seen these fungi before, and it would be pretty easy to see them. Sometimes, other times, maybe not so much. But either way, you can find them almost around the entire world because they're just so common and hyper diverse. There's like hundreds of species. And the way you can, I guess, just the quick and narrow of how to identify these fungi just by looking at them is they have these hard black fruiting bodies. We call them stromata, and they can seem sort of like fingers, like dead man's fingers, the famous Xylaria polymorpha, or they can be these sort of crust-like forms, or maybe they look like these like tiny balls on a log. Like these would be a lot harder to see, but they're out there. And if you're looking at rotting wood, you notice these sort of black blotches. That could be signs that a Xylarialian fungus is there. So um, for this presentation, there's a couple definitions I need to lay out because I'm doing a phylogeographical study which incorporates first biogeography, and this is studying the geographical distributions of species. So just knowing where in the world certain species live, maybe how they live there, the adaptations that make colonization successful. And I also used this is an basically like a metric to determine the similarity between two organisms that are in different areas of the world. It's a biogeographical signal, and that's just looking at the genetic similarity of two organisms that are in different biogeographical realms, we call it. And these have been defined in numerous sources, but just as an example, this mirrors the guidelines set in my thesis um, pretty to the point. This is by the World Wildlife Federation, and it shows various areas such as the Nearctic, the Palearctic, the Neotropical. So my study area included a small island in French Polynesia known as Moorea, and this is where my master's advisor Todd did a biodiversity project he participated in known as the Biocode project. And this was basically an effort to fully catalog the biodiversity of a tropical island ecosystem. And this would be easier than most islands because Moorea is actually really small. It's about half size of Kauai, which is the island right below that, as you can see, and that is a small island within itself. So I wanted to study Xylarialian fungi, not just in the small French Polynesian islands, but I wanted to branch out to the Hawaiian Islands as well. So I've gotten specimens from Kauai, Hawaii, and Oahu, those three islands shown there. And I've obtained these specimens through various biodiversity projects that unfortunately I was not a part of during the field sampling, but luckily I was able to access these specimens and uh, analyze their DNA. So my big question for the research was which biogeographical realm has fungi, the Xylariales, most closely related to the Polynesian types? So looking at the area of Polynesia, which biogeographical realm had the most genetically similar fungi? Was it the Nearctic, the Australasian, the Afrotropical? Right, that's the question that I was asking, and that would then lead to potential methods of dispersal for these fungi, and also possibly tracking evolutionary histories across continents. So my first objective was to determine biogeographical signals for 82 specimens of Polynesian Xylarialian fungi, and this is done through a genetic analysis and a bit of computer work as well, which I'll get into. And then the second objective was to, for a little more detail in terms of evolutionary history, was to put these fungi on a cladogram, which is kind of like a family tree, but at the tips of the branches, there's biological species or isolates of a particular species, and you can see how how they're related to one another using that branching diagram, and comparing that to fungi that have been sampled in the past, but from other biogeographical realms. So there was this huge phylogenetic study in 2016 by Uren et al. And they have sort of like the reference sequences that I used for those other biogeographical realms in the study as a comparison to the Polynesian material. So, as any genetic analysis goes, you have your DNA, right? That needs to be extracted, but you also need to amplify the certain genes that you're looking for. You are typically looking for certain genes, and what I was focusing on was, well, one of them was ITS, and it's a great sort of barcode um, for a species of fungi or a species in general because it's something you can find in just about all life. It highly varies among species, so it's good to sort of differentiate them based on the arrangement of nucleotides, and it's something that people have already sequenced a bunch and has a lot of documentation in the database. There's a sort of like this block between the ITS1 and ITS2 spacers called the 5.8S gene, and that one's highly um, not it's not variable at all. So I didn't put it in my research. I did not amplify that gene because it would just be just sort of redundant. Wouldn't give me much information. But those IETS 1 and 2 spacers, those are the things that I'm looking for. Um, and before I go any further, I want to share that there were many more genes that I was willing to amplify with this, but um, these are old specimens, and um, the Xylariales are kind of infamously difficult to extract DNA from, even. So, um, you know, not a lot of success. But ITS was the gene that was just kind of that's where the money was. So my first objective, the biogeographical signaling, started with um, taking off from the sequencing. I got my DNA samples back. I got them sequenced at the University of Wisconsin-Madison. They have a nice Illumina sequencing machine there. They returned the sequences to me, and what I did was I put those sequences through a blast search, trying to find the best matching sequence to my Polynesian fungi. And when I found the best matching sequence, I looked, okay, well, where is it from? And let's say in this case, the best match is from Japan. And as the region with which Japan is within has defined this biogeographical region as Palearctic. So that would mean that the Polynesian fungus that I've put into the blast search would have, according to this analysis, an Eastern Palearctic affinity, meaning that it is most closely related to an Eastern Palearctic species. And I did this with all the samples that I possibly could. For the Morea data, it was very lacking. There's supposed to be 35 representatives here, but there's only 9 because the specimens were like 2 decades old at this point, and getting DNA out of them even was just such a pain. So what it shows is predominant Indo-Malayan signals. So this is the Indo-Malaya biogeographical realm here, and that's what it shows for Morea. And that gives the idea that Morea xylariales are most closely related to Indo-Malayan types, but with such a small sample size, I cannot be certain about that. However, with the Hawaiian fungi, they were a lot more successful um, in terms of sequencing, and I had an additional um, 47 um, 47 specimens for the Hawaiian Islands. So it was more than Morea, but also a lot more successful overall. And it shows predominantly in the neotropical areas. In the neotropical area and the Indo-Malayan area is where the greatest biogeographical affinities were. So it's surmising that the Hawaiian Islands have xylarialian fungi most closely related to Indo-Malayan and neotropical types. So to take away from this analysis is that the relatedness, which I've stated before, and we cannot be certain about the Morea types because of the small sample size. Now, these fungi can disperse in many ways. They can be transported via living or dead plant material because they can be decomposers, but they can also be endophytes, so they can live asymptomatically in living plant tissue. They can also live as endophytes in marine organisms like sponges and algae. So they have a lot of ways to get around. On top of that, they have the wind dispersed spores. So dispersal is probably a bunch of factors leading up to the colonization of these islands. I don't think it's co-dispersal with their plant hosts, like live plant hosts, like an entire like tree or something like that, unless it's horticultural imports or if there are any fungi associated with the seeds, then maybe both the plant and the fungus have colonized. But I don't believe that's likely. There's very few species that associate with seeds, to our knowledge. And I believe that since Indo-Malayan and Neotropical plants um, were were outlined in a biogeographical study of Hawaiian angiosperms by Price and Wagner 2018. I think that having those plants on the Hawaiian Islands has allowed the fungi to colonize those islands better because there are hosts there are hosts there that they're already kind of familiar with. So it gives them a little more leeway than other fungi from other biogeographical realms to sort of colonize. And last thing is sort of a caveat is that there's kind of a regional bias towards the Indo-Malayan and Neotropical types for my results because the Xylarialian fungi have they have disproportionately high documentation in these areas because that's where these sorts of experts of xylarialian fungi that are alive today, that's where they currently reside. So it is possible that because they do so much sequencing in those areas that the overflow of data is just kind of pointing to those regions when it may not really be the case. It's mainly a data availability issue, is what I'm saying. As for the second objective, this is where I show my phylogenetic tree or area cladogram. And by area cladogram, I mean a cladogram that takes into account the geographical localities from which each fungus was sampled from, right? So the original database that I was using included my 82 specimens and those reference sequences I mentioned earlier, totaling to 845 specimens or leaves, we call them on this tree. And after much whittling down into something that kind of made sense, I was left with only 78 leaves. And oh, that's because not all of my 82 samples had amplified. So looking at this tree, there's there's a lot of different things going on. I was able to pick out a few interesting things, which it overall shows that the Polynesian Xylariales have variable biogeographical signals. Again, um, this is more certain in the Hawaiian data because a lot more of those specimens were successfully sequenced. But you can see there's two samples of Biscaynoya. The one in gray is Polynesian. This one is from Spain. So Western Palearctic. You have Nemania bipapillata, which is a Polynesian here in the gray. And then you have two from the Indo-Malayan area, Indo-Malaya region. Crachemia clavis, most closely related to Crachemia adusta from Germany. So different areas of Peru, different areas of the world, which, it's something that I would expect from fungi that are so well distributed. But it's, something interesting also for such an isolated area of the world, the Hawaiian Islands, that there's these sorts of trends across different areas of the world, all converging onto the small archipelago. There's some important limitations here, such as the ITS. The ITS region by itself isn't great on its own for phylogenetic analyses. It doesn't give you a whole lot of certainty, and that's where you get a lot of low bootstrap values. They're basically confidence values, and you didn't see a lot of those on the area cladogram because most of them were below 50, and that is basically there's no support for that particular arrangement. There was also a lot of lack of data in terms of, what species is this? Where was it from? And so that's harkening back to people who go out in the field and collect these things. Sometimes they don't take all the data that they need. And, we need to do a better job of that if we want to do biogeographical studies in the future for these fungi. And then the regional biases, as I mentioned earlier, with the Indo-Malayan and Neotropical areas. So that was my master's thesis project, and the work has been finished, but I'm looking to publish that paper soon in a Mycology or something like that in a scientific journal. But now I'd like to share with you a project that I have that I've studied with an undergrad that was willing to do research with me and was overseen by entomologist and amazing artist Dr. Barrett Klein. And this was sort of like all of us converging on this one project, just kind of doing just kind of putting in a little bit of everything that we like. So I'm really interested in Laboulbenian fungi, and I will tell you about what those are, but they're really cool. Zachary wanted some research experience, and he was interested by this by the idea of this project. He came up with his own question, his research question. And Dr. Klein is really interested in insect behavior. So he had a lot of fun helping us set up the observation experiments for this particular project. So the Laboulbeniales are a microscopic fungi that you can only find on the exoskeleton of arthropods, usually beetles, right? But they can be found on many other insects and millipedes and mites and things like that. So like the Xylariales, they're quite they're quite well distributed across the globe, but people rarely hear them, let alone see them themselves. So we know very little about these fungi, even things of like how do they affect their host. And right here we have a study by Conrad et al. 2014, and it was looking at Laboulbenian infections on ant colonies. Right, there were some individuals that had very low infection rates, so very few thalli or fungal fruiting bodies on them, but some of them had a ton on them, made them look all fuzzy like. And what they found was that between these two groups, you had the highly infected individuals were starting to die off quicker than those that were less that had less fungi on them when they were deprived of nutrients. So it seems like a bit of a parasitism there. But if they're healthy and they're exposed to a lethal fungus, such as a metarhizium, which would kill the ants, then it could be considered sort of a mutualism because they have all these fungi on them and they start brushing them off like crazy. They increase their sanitary behaviors, and as a result, they're sort of dispensing of those lethal fungus spores off of them, and they don't get killed by the metarhizium. So they actually started surviving better when they were healthy after being infected with a ton of labules, is kind of the colloquial name for them. In addition, when they have a ton of labules on them, they also experience upregulation of certain immune system genes. So it's almost like they're being prepped for infection if it does happen, by like a different fungus that really wants to, um, really wants to hurt the ants. So the first objective of this project was to compare the quantity of food consumed by beetles that were not infected versus those that were infected by Laboulbeniales to see if they ate more or less than the control group. Also, we wanted to observe the grooming behaviors, specifically the rates at which they would groom and how long they would groom. So basically, how many times would they just go into a session of like, okay, I'm grooming myself, and how long would each session be between the two treatment groups? So first, we needed to collect our beetles. We went to a nearby marsh that's very close to the university. It's a walking distance. It's called Myrick Park, and the nature place there is kind of like, kind of like the place that organizes the marsh. They organize efforts for research, and they allowed us to um, collect beetles from there. So we're very grateful for uh, having them, for them letting us collect there. It's a very great site, and we basically went along the shorelines of this marsh area and started corralling all these beetles into into these into these little bottles, right? And the beetles we were aiming for were Carabidae beetles, family Carabidae, commonly known as ground beetles. And this is important because ground beetles are the predominant beetles that are infected by labels. They're the ones that are most commonly infected of basically all beetle families. So we thought best chance of getting infected insects would be to go for beetles, specifically Carabidae. And we did collections at these three sites where the red dots are located, and we brought them back to the lab to scan them for infection. And here's an example of an infected beetle here. This one's dead. This one's from a project I did in my undergrad, and we did not kill the beetles. We kept them alive for observation of their behavior, of course. So we put them under a dissecting scope, put them in each one of these like trays to make sure they weren't cross contaminating each other, looked for infection, and sometimes we had unexpected hitchhikers as well, such as a sporadic mite here, which actually appeared on almost all of the infected beetles. So whatever disparities in the behaviors between the two groups um, may also be caused by these mites. But the data is kind of to be determined on this. So the filming setup was just kind of these security cameras, part of a LoRax system that we pointed down um, onto these beetle enclosures, and it's just like a regular like security camera system, very DIY setup using ring stands to point these cameras down at the beetles, and just little acrylic cubes to place them in. We gave them food. We made sure to weigh that and the beetles before and after, as well as this watering gel here. It's a solid form of hydration for these beetles, so it's a lot less messy than liquid water. And we used a 12-hour dark cycle to acclimate these beetles to a sort of a day and night time environment, just to keep it consistent with what they were experiencing outside. So we have very preliminary results at the moment. We haven't finished going through all the footage, but what we see so far is that the infected beetles consumed more milligrams of food. Milligrams is how we measured the food than the control group. So it is possible that these beetles, because they have these fungi on them, they're starting to eat more, maybe because these fungi are depriving them of nutrients through absorption of maybe their cuticle fats, or maybe the beetles are just brushing themselves so much that they're burning more energy. Though we found in our grooming segment that the duration of grooming was not significantly different between the treatment groups. And this is possibly because that the beetles that we were able to capture were not highly infected. They had like 1 to 3 thalli on them, and it wouldn't be a whole lot even for a beetle's standards. But it's important to look at those minute differences, even because people who are looking to study the behavior of these beetles in the future need to be wary that if just these few of fungi, just like 1 or 3 thalli on the fungi, can alter their behavior, then that's something to look out for. And again, those phoretic mites as well could have contributed to changes in behavior or food intake. Okay, thank you everyone so much for listening to this presentation. These are my acknowledgements for the Xylariales project, my field collectors, undergraduate researchers, mentors, and thesis committee members, as well as the agencies that have funded me and given me resources throughout this project. And a special thank you to the Minnesota Mycological Society for the Scholarship Award and for allowing me to present my work today. And additional acknowledgements for my much smaller Laboulbenian project, the College of Science and Health, the fellowship committee of the dean that gave Zachary the scholarship award for him to conduct this project over the summer, given the proper funding, Myrick Park and the Nature Place for letting us sample, and also a friend of mine, Lauren Ray, who has helped Zachary analyze the footage of these beetles, which is something she's apparently done in the past, which we really appreciate that. And I am just so thankful for all the people that I've worked with on these projects. Can't thank him enough, can't give him enough credit. Okay, thank you, everyone.
Well, we could move into, yep, lots of claps. Thank you so much, Michael. It's great to have you here tonight. I am going to then introduce our next speaker, who is Jason. Jason, is it Raiti?
That's correct.
Perfect. Yeah, I met Jason at the Santa Cruz Fungus Festival. And learned about all the different things he was doing. I encourage you to check out his website, the Sclerotia Project, which I think is pretty exciting. Kind of paying it forward to the next generation of people who are studying mushrooms. Jason is currently pursuing a graduate degree in botany at the University of Wisconsin-Madison, where he studies evolutionary morphology of Amanita spores in the Pringle Lab. His research integrates ecology, phylogenetics, and computational analysis with a strong focus on science communication and fungal conservation. He also serves as a board member for the Madison Mycological Society, where he runs their speaker series. And prior to joining UW-Madison, he served as a board member of the Central Texas Mycological Society, leading hands-on workshops in low-tech mushroom cultivation and wild-to-agar cloning. His work has included cultivating bioluminescent fungi in Brazil, co-authoring a paper on robotic water sampling for IROS, and contributing to a NASA-affiliated robots project, including an autonomous lunar rover and a space trash cleaning robot. Outside of academia, he enjoys doing anything outdoors, hiking, foraging, surfing, climbing, running, but also loves to play guitar and make any kind of art or poetry. Jason believes that creativity and science are deeply intertwined and strives to bring a sense of wonder and playfulness to everything he does. I think the lecture I saw you do was Mushrooms in Art, and you led us in a song with a ukulele at the end, which was the perfect example of combining science and art. So yeah, thank you. Thank you for being here tonight.
All right. Well, thank you, Kathy. I appreciate that. I forgot about the song, but maybe another time I could come back and I'll deliver it for the Minnesota Mycological Society as well. All right. Well, first, let me start with saying a huge thank you to the Minnesota Mycological Society for putting this together and for selecting me for this wonderful scholarship. I really appreciate it. As a grad student, it makes a huge difference to me, and it's really just, I started in mycology as an amateur mycologist working in the San Antonio Mycological Society, and I fell in love with it through that work. And it's really special to continue to try and be a part of these communities. And yeah, so I'm excited to speak with you guys here. You're going to see what is essentially my thesis defense for my master's degree. So 2 years of work. I'm going to try and summarize it here in 30 minutes or less for you. And this will be fun for everyone, I think, because it's very interesting. If you pay attention to the very end, there will be a pop quiz. So, keep your eyes open. And I'm excited because, well, I was stressing about this presentation like you can't imagine. And now I get to do it for fun. So let's get into it. Jason, Kathy, thank you for the wonderful introduction. I study mycology and I would like to become a mycologist, you might say, a scientist. But in my free time, I'm just a regular, person who enjoys the outdoors and hanging out with my friends and doing things like that. But this research is more than just me. I want to take a second here to acknowledge all of the people who put work into this and who I'm speaking on the behalf of. So Corbin, Brian, Sydney, Sheldon, Carson, Carrie, Jacob, Ricardo, Denny, and Anne. Anne is my advisor and PI. And the rest of these folks either helped with data collection, data analysis. There's some really fancy math behind this project. There's a lot of really intense phylogenetics behind this project. So this is the work of a whole group of people, not just myself. All right. So today we're going to be talking about what influences spore shape. Essentially, the question that I have is what shapes spore shape, specifically in the genus Amanita? And to answer this question and to try and give you a hook, I'll first give you a primer on shape in Amanita. I'm going to tell you a little story. I'll give you a review of the background literature and then what hypotheses we draw from that. And then we're going to talk about something called morphometrics and the methods we use to answer this question. I'll present my results, give you the conclusions, give you a little bit of the theory that we're developing. And unfortunately, since this is not in person, I can't give you free stickers, but there will still be a quiz. Okay, so what shapes spores? Well, if you've never thought about shape in nature, now is a great time to look outside your window and think about it. So next time you're walking around the woods, I want you to look at the different leaves that you see around you. I want you to look at the kind of textures you see on tree bark, or the different kinds of wings that bugs have, or the mouth parts of birds, famously used by Darwin to describe evolutionary change over time. So shape is extremely important for many reasons. In ecology, it's important because we often talk about this phrase form equals function. And in evolution, shape and morphology are the basis of evolutionary theory in a very literal sense. Now, spore shape, on the other hand, unlike leaves or beaks or wings, is very, very understudied. We know very little about spore shape. So here's a picture of the diversity of spore shape. You can see in the center here are a variety of different kinds of mushroom spores. Top right, we have some pollen. Left side, you have an extremely small seed. And then in the bottom right, you have an extremely large spore. This might be the largest spore in the fungal kingdom, at least the largest that I'm aware of. So there's quite a large diversity, but you'll notice that these are all very, very small objects. They're microscopic. But even still, you see ornamentation, warts on some. You see some are very elongate, is a term that you'll want to remember. And some are kind of odd shaped, triangular, pyramidal, so on and so forth. All right, so let's talk about Amanita. Everybody knows Amanita mushrooms, even if they don't know that they know Amanita mushrooms. Amanita muscaria is our mushroom that is like the quintessential mushroom, right? So that's like the red and white spots mushroom. Now, I like to say that Amanita is a model genus because Amanita is first and foremost like the panda of mushrooms. It is extremely charismatic, but it's also highly widespread and very interesting ecologically. So it's a model genus because it allows us to take a look at this particular dichotomy here between ectomycorrhizal and saprotrophic fungi. So if you never heard these terms before, ectomycorrhizal essentially means that a fungus survives by trading nutrients that it gets from the soil to a plant for that plant's sugar. And saprotrophic means that it just eats organic material in the soil, breaking it down. So in Amanita, there's a single origin of ectomycorrhizal symbiosis, which makes it a good system for studying this. It has both. All right, so essentially the story that I'm going to tell you here is that mycology is behind the curve. So remember, you are walking around the woods. Now imagine you are a botanist walking around the woods in, say, Bhutan, and you run across this really cool looking, I think this is an orchid species, and you notice that these orchid seeds are extremely small on this mysterious orchid. And if you were up to date on your botanical literature, you would know that small dust-like seeds are an adaptation for establishment in high resource environments, right? If you have a very small seed, you're not packing a lot of resources with you, so you're expecting to go somewhere where there's a lot already there. So if we compare that to spores and fungi, essentially we have no idea. So let's cover what we do know about spores and fungi, the current literature. All right, so one of the first ways that people have tried to understand spore shape and ecology is through climate. So some people say that really large spores will be found in warm, wet climates. Really large spores are more likely to be ectomycorrhizal. They are more likely to reproduce early in the season. They're from continental regions, and they have larger fruiting bodies, reproductive bodies. Basically, the mushrooms themselves are bigger. Vice versa, you get the opposite with small spores. They're in colder places, they're saprotrophic, they reproduce late in the season, they're from coastal areas, and they're from smaller mushrooms. On the other hand, on the shape axis of variation, very elongate, which is this bottle-like shape that you see in the center of your screen. Very elongate spores are more likely to be saprotrophic, whereas on the other hand, globose or beach ball shaped spores on the right side are more likely to be ectomycorrhizal. And some folks say that it's a little bit of both, where ectomycorrhizal fungi are large and spherical, and large spores tend to be spherical. And on the flip side, saprotrophic fungi tend to be small and elongate. And of course, it wouldn't be science if you didn't have some folks saying that it's none of the above, or it's completely different. And some say that ectomycorrhizal and saprotrophic fungi do not differ in shape or size, and that these traits are actually strongly phylogenetically determined. So that means essentially the trait is predicted by your evolutionary history more than your environment. If your parents had this trait, you're likely to have this trait in the crudest sense of that. All right, so what do we see here? Essentially, there's no consensus. We have no idea what it really is. It seems like people have done targeted studies looking at very specific factors, but there's no overarching narrative that we can glean from this. So what our hypothesis for this project is essentially there will be no single factor influencing spore shape. It seems pretty clear that spore shape, although it may be relatively simple to describe sometimes, will be very complicated in terms of what is influencing it. So we're going to take a look at three things here. Trophic mode means ectomycorrhizal or saprotrophic. Essentially, what do these fungi eat in nature? We're going to look at phylogenetic factors, so whether or not it's your ancestry that determines your trait, or if it's something in the environment that is putting a selective pressure on you. And then we're going to look at that environment in finer grain. So we'll look at factors like precipitation, climate, and geography. Okay. So quickly, I'll tell you how we did this. For a species to exist, it has to be described to science. This is just how it happens, how the sausage gets made, and it's a very tedious process. But this has been going on for many, many years, and although we have just scratched the surface, we have a lot of data, especially in Amanita. Amanita has been well known for a long time, and so there's a lot of what's known as taxonomic literature, species descriptions. So here's an example. On the right side, you'll see an illustration. Typically, it's a painting or a hand drawing, or these days it would be like some high quality photographs of the mushroom, the hyphae, and the spores. And on the left side, you can see here, this is a sample of the metadata that would come with one of these descriptions. It usually is more standardized these days, but in essence, it just describes things qualitatively often that whoever's describing this species has noticed about it. All right, so the way that we've done this project is essentially we want to get the best data we can on the shapes of spores. So we extracted from this taxonomic literature spore shapes and metadata. This metadata and spore shape database we compiled all together. We ended up with over 200 samples from the taxonomic literature. And that data set of 200 samples we took and digitized. Essentially, each one of those spore shapes, it's a 2D outline. We took those 2D outlines from documents and we made them into digital form that we could manipulate. And so in the digital form, you need to normalize these spore shapes. So that means each spore has what's known as a hilar appendix. It's where it attaches to the basidia. I know I'm saying a lot of crazy words here, but essentially it's a nipple that allows the spore to attach to the gill and the place where these spores are launched from. If you've never seen one of these spores launched off of a gill, I'll tell you, look it up. It is very cool. It's called ballistospore. So we also smoothed the outline. We standardized the size of the image and centered the images. So all of this is important because this enables us to get this center image here, which is all of our spores layered on top of each other. And then we can use this data set for what's known as morphometrics. Now, morphometrics is a huge field into its own right, and I will briefly describe it here. So I'm going to describe it using my friend Corbin. Corbin, he likes fishing. So Corbin, he's fishing, he catches a fish. One of these fish is very, very long, right? This is an oarfish. He goes fishing again and he catches a sunfish. This fish is very, very circular, very wide, you might call it. Now, Corbin is a scientist, and Corbin wants to understand the shapes of these fish in a scientific way, not just to say, well, one fish I caught was very long, one fish I caught was very circular or wide. So what does Corbin do? He takes these fish shapes and he takes the outlines from these fish shapes and he essentially uses some fancy math that enables you to describe the ways that these outlines vary in comparison to each other. And that is morphometrics. Essentially, it allows you to create axes of variation that aren't necessarily things that we understand intuitively as axes, like height and width. This would be more like bigness or roundish wideness or so on and so forth, right? So the point of this is the axes of variation that I'm going to be talking about from here on out are not necessarily real axes, though we intuitively can describe them in ways that can only be quantified with morphometrics. So for these spores, you will see shortly that this axis is going to be along this particular line here. Not length, not width, not size per se, but somewhere in between. So what characterizes spore shape? In Amanita, something interesting we find is that spore shape is remarkably simple, right? So in nature, go anywhere in nature, look at anything in nature, and you find an immense diversity of shapes. That is not the case for Amanita spores. Amanita spores, we see a lot of diversity, but in a very constrained way. Almost all Amanita spores are along this elongate to globose axis. And that's what this plot in the top right is showing here. It's just showing the percentage of variation based on axis of variation, principal component. So what you need to know from this slide is these spores are essentially all either somewhere on the elongate side or somewhere on the globose side or somewhere in the middle. So first things first, we wanted to take a look at volume and this elongate to globose axis. So volume is important because our way of measuring spores is inherently two-dimensional and spores are three-dimensional shapes. Volume helps us capture that. So taking a look at this graph here, you'll see that shape and volume are highly linked. So elongate spores all appear to be very, very small in size, in volume, and globose spores can be pretty much any volume. Something notable here, though, is that this top left quadrant of what could be called morphospace is completely empty. There's nothing there, right? And when you find essentially a trait in nature where there's some possibility of the trait existing, but there's none found in nature, it's usually a sign of some kind of biomechanical constraint. So, what variables influence the simple shapes of Amanita spores? Well, let's take a look at trophic mode first. Just a reminder here, we have ectomycorrhizal, saprotrophic categories. I'm also throwing in here an unknown category. I'm not going to give you all the background here, but recent genomic evidence is essentially complicating our understanding of a small subset of Amanita mushrooms. They may be somewhere in between ectomycorrhizal and saprotrophic. They're not necessarily distinct categories, but maybe more of a spectrum. So when we take a look at this analysis, what we first find is a slight naive, significant difference between these groups, right? However, we wanted to go further and we looked at these groups when accounting for their phylogenetic history. So essentially, when you look at how different spores are and you take into account who their parents were, right, from a species level. So we're talking about like thousands and thousands of years of parentage. What we find is that parentage is a better predictor than necessarily which of these trophic mode groups. So essentially, trophic mode can't be disentangled from who your parents are. So next, we want to take a look at shared ancestry. This is just looking at who your parents are alone. And interestingly, we find that there's no pattern based on shared ancestry. The easiest way to intuitively show this is, if you look at this figure here on the right, the blue tips are very, very globospores. Each of these tips represent a species. So the blue ones are very, very globospores, and the red ones are very elongate spores. And you'll notice that blue tips and red tips, they are all over this tree. And oftentimes, in some places, you'll find a blue and a red or a yellow tip right next to each other. That's very strong evidence that this trait is evolving unpredictably and not gradually. Essentially, this trait is convergently evolving over and over. So, what variable influences the simple shapes of Amanita spores? Well, last hypothesis from the literature, it's climate. So let's take a look at climate. We took data from NOAA and use these Köppen climate zones. These are very standard climate zones. Also, shout out to NOAA. We couldn't do this project without NOAA data. It's a real shame what's going on with them. I won't get into it with you guys, but supporting environmental science is huge, and we should continue to fund and, yeah, just generally not dismantle NOAA if at all possible. My personal opinion. All right. So what we find is that spore shape differs strongly by climate. It seems to correlate very strongly. So in particular, spore shape and 30-day precipitation are highly correlated. 30-day precipitation, I'll tell you the context here. Essentially, when these mushrooms, each of these species were described, there's a particular collection that is the holotype, that is the example of that species by which all other specimens will be compared to. And we looked at the 30 days of precipitation prior to the collection of that original collection. So this is supposed to capture some kind of like local conditions. And we find that these local conditions are actually very, very important. Also, the climate groups, local conditions are cool, but evolution happens on the time scale of like millions and millions of years. So we also wanted to look at like broad climate trends. And we find that these climate groups are also significantly related to spore shape. So just to describe this here to you in words, globospores tend to be found in more continental areas and in drier local conditions, whereas elongate spores are found in temperate regions and in wetter conditions. Now, something interesting about this climate grouping is taking a look at this geographically, we find that all of these really, really elongate spores in the temperate regions are all from a similar place. They're all from this coastal region of the southeastern United States, all of these species. And I took a look at these species ranges, which what I noticed just qualitatively is, although some of them extend up as far as New York on the East Coast, all of these guys, their ranges are concentrated along the coastline. They don't extend very far inland. So that's interesting because that is one of those hypotheses from the literature. Also, this is not a monophyletic group, right? This is a group of fungi that are not related to each other. They are all Amanita, but they're not all from the same part of the Amanita family tree. And yet they've evolved the same shape and they're found in the same place now. So just crossing our T's and dotting our I's, essentially for trophic mode, we find strong evolutionary signal that makes it hard for us to determine if trophic mode is actually a predictive trait or a predictor of this trait. Well, we don't find that for climate or precipitation. Essentially, the way that spore shape appears to be responding to precipitation and climate is completely independent of their ancestry, their evolutionary ancestry. And because climate and precipitation are related, we wanted to essentially compare them together to say, are these two models better separate or together? And if they are better separate, which one's the best? And it seems that spore shape in particular is most related to very localized conditions. So we're talking maybe even less than 30-day precipitation. That's a speculation, but it seems that rather than broad climate trends, it is these very localized conditions that are the most important for spore shape. So what variable is the most influential? Well, it's complicated, and that's okay. So let's review what I just told you here. Essentially, Amanita spore shapes are simple, but the factors that influence them are not. Amanita spore shape is along this axis. We have globospores, we have elongate spores, we have some in the middle, but pretty much all of these spores that we observe in nature are somewhere along this spectrum. Now, in terms of shape and size, we find that globospores, large spores are constrained in shape to being globose, and small spores are unconstrained in shape, whereas elongate spores are pretty much all very, very small, and globospores can be um, any size. These elongate spores are found in high precipitation environments, and the globospores are in low precipitation environments. And elongate spores are in temperate climates, whereas globospores are more likely to be in continental climates. Overall, we find that small elongate spores are constrained together, right? And that they're found in the coastal regions of the United States. And we find that in terms of evolutionary history, spore size and shape as traits are not explained by ancestry. They don't appear to be evolving randomly or gradually based on parentage. So in other words, this is a trait that is under active selection, something that is determining the fate of spores. And I think that the main finding here is that spore shape is remarkably labile. Can you think of another trait that is convergently evolved over and over and over again, even inside of like a family of related organisms? I'm sure there are other examples, but this is a pretty remarkable finding, I think, that spore shape seems to rapidly transition between these two extreme states. So, if evolution keeps arriving at similar spore shapes, what problem are the spore shapes solving? Well, this is a difficult question, and this is essentially the ecologist's version of saying, what the heck is going on? So, the answer that I am going to try and give you today is that it's all about spore biophysics. Now, I talked a little bit about ballistospores before. Here's a crash course. Basidiomycete fungi, so that's your club fungi, all have this launch mechanism. Essentially, the spore sits on a little tower, a bubble of water grows on the nipple of the spore, this little bit that extends off to the side of it. That bubble of water collapses onto the spore once it reaches a critical size, and the movement of that water shoots the spore off into the space between gills at like G forces equal to a rocket launch. Something insane. It is, I mean, it's an incredibly crazy system for moving a spore like half a millimeter, basically. But all of this is to say that spore shape is clearly important for something that is essential for the fungal life cycle, which is getting your spores off of the gill and into the air. And spore shape is also important for dispersal. So once, there you go, a video of spores dispersing. Once those spores get into the air, they have to first escape the region directly around the mushroom. So if you didn't know this, essentially air sticks to things. So the wind, if there is wind, moves more slowly next to an object. And then there's atmospheric transport. These spores are interacting with turbulence with whatever air currents there are. And then they have morphological filtering. This is where like small differences in spore traits probably make for big differences in survival outcomes in the long run. And the thing that's selecting those spores, which ones survive and which ones don't, are probably environmental gates. So that would be like humidity. Essentially, if it's super dry, a spore will desiccate and it will die before it ever even reaches the ground. On the flip side, if it's very humid, Basidiomycete spores, as we were talking about, are coated in chemicals that allow water droplets to condense on them. Well, there's a lot of research that shows that Basidiomycete spores in particular might be essentially seeding the rain because water droplets condense on them and then they'll rain out of the sky. And UV is another serious risk of spores, and we see in other genera highly pigmented spores. So, last thing I'm going to cover here, I'm running low on time. Essentially, spore biophysics. Spores exist at Stokes flow physics regimes. This is just super small scales. When you are super, super small, the air molecules relative to you are actually rather big, and they are very grippy. You can just imagine what it would be like to swim through corn syrup. And here's a demonstration of Stokes flow. So essentially what that shows you is that whereas in non-Stokes flow, in a fluid, if you mix it up, it's impossible to unmix it. In Stokes flow, you can perfectly unmix a fluid by just reversing your motion. Essentially, the point here is that there's unintuitive side effects at being in this strange physics regime. So terminal velocity, I think, is essentially the answer to what problem spore shape is solving. There's a bunch of assumptions that I made to create these terminal velocity models. But long story short, what we find is that across the genus Amanita, the terminal velocity based on spore shape varies about fivefold, and very elongate spores are associated with much slower terminal velocities. So this small number here means that your terminal velocity is slower, whereas the large number is a faster terminal velocity. Essentially, if you elongate, your spore is likely to fall at a much slower speed than if you're globose. All right, so that is pretty much it. Here's the pop quiz. I will, let me see if I can see this chat here. Okay, so looking at these guys here, a feather falling in air with its broadside orientation facing down. What direction do you think this feather will fall? Kathy, you got an answer?
I don't.
Well, I'll give you a hint. It'll fall straight down. All right. All right. Tip down. It'll fall. Eventually it'll stabilize with its broadest face facing the direction of travel. Right. That's the most stable one. Now, what if this feather was falling starting at an angle? Which way would this feather fall? Anyone? Be brave. To the right, and you would be sort of correct. At first, it would fall to the right, and then over time, if it fell for long enough, its orientation would stabilize, and it would begin to fall with its broad face down. You could think of like if you ever dropped a piece of paper, you see it swing back and forth. That swinging back and forth is it stabilizing its falling. Now, what if we took a spore and did the same experiment? A microscopic spore falling with its broad face down, how would this spore fall? Be brave.
Same as the feather, straight down?
That's right. Good job. If I had a sticker, I'd give you one right now. All right. Last one. Now, what if this spore was angled sideways? What direction would it fall?
Same as the feather, or same as the feather again, sideways first, right?
Sideways first, and you would be wrong. Oh, that was a trick question. I'm sorry. So something cool about Stokes flow and spore shape is that the elongate spores, basically having an irregular shape um, in the Stokes flow, means that you fall in an unpredictable way, right? So being in Stokes flow, you whatever orientation you have when you're falling, there's no oscillations. You just stay in that orientation, and because your orientation does not normalize, does not stabilize to the most balanced way to fall, you actually will probably end up falling somewhat to the side, some random side actually. And this probably has something to do with like a spore just released from a mushroom falling to the side is more likely to escape the still air that's directly surrounding the mushroom, potentially, whatever grass or leaf litter is around it as well. So all of this is to say that spores have shapes. Those shapes are relevant and they deserve to be studied in terms of their physics. So spore dispersal, biophysics, important. Nobody studies it. Basically, someone should look into this. Okay, the story is mycology is catching up. Imagine you're a mycologist, you're walking around my home state of Florida, you find this mushroom, and you notice that this unknown species of mushroom has very, very small elongate spores. You would be able to say with some confidence this species is likely adapted for fruiting or in relatively high precipitation environments. And for future research, if any of you guys, amateurs, there's a lot of cool experiments you could do here that really are low tech. And, all you need is a microscope and some elbow grease and some grit. But someone ought to look into interspecies variation in spore shapes, how phenology affects spore shapes, and then, of course, these ballistospore and dispersal biophysics. This could be as easy as taking two mushroom caps with different shaped spores of approximately the same volume and trying to measure how far those spores disperse just based on the variation in shape. And that's it. That's my presentation. I appreciate you guys for taking the time to be here today. I'm a little bit over time, so I don't know if we'll have time for questions, but I am really grateful to be here. And yeah, thank you so much.
Thank you. We totally have time for questions, and I see some in the chat from Randy. So, Randy, I wondered if you just want to unmute. You had a question earlier about the saprobic versus ectomycorrhizal.
Yeah, I'm wondering with the Amanita species, are they exclusively saprophytes or ectomycorrhizal, or is there more of a continuum between the two, depending
So
where
that
they
is that is a great question. Thank you, Randy, for bringing that up. So I mentioned this very briefly, but I included an unknown group in my analysis. That's essentially because I did a bunch of genomic work for this project, and that genomic work essentially is starting to reveal that there probably is a spectrum of what some people call lifestyles, but I try and call trophic modes, right? In the past, people have considered fungi as in these discrete categories. You're either ectomycorrhizal or you're a saprotroph. I think more and more people are coming to accept that some fungi fit neatly into those categories, and then others are probably in an intermediary state where they can sometimes associate with plants, they can sometimes live freely, and just eat organic material in the soil, and maybe why and when they do those things can depend on the context that they find themselves in.
Thank
Any
you,
other
Randy.
questions?
Yeah, Randy, you had one other question.
Yeah, I was wondering about gill spacing and gilled fungi. That can vary quite a bit. Does that have an influence on spore shape and morphology?
Okay, another great question. So for ballistospore launch, let's see if I can find this slide again. For ballistospore launch, right? Okay, let's go here. So this is our ballistospore. This picture on the right, that vertical surface there, that is the gill surface. And then those things that are shooting off and then falling down, those are the spores. Now, this whole Rube Goldberg machine, this is just to get a spore half the distance of a gill space, right? Half the distance. It's a crazy system to go a fraction of a centimeter, essentially, in most cases. Now, there are mushrooms that have very widely spaced gills, and I've read papers that essentially argue that the gill spacing tends to be tweaked just perfectly, such that the launch force from the bubble collapsing onto the spore gets them exactly halfway. So I think that there is something going on with gill spacing. The truth is, nobody's really looked into it in a rigorous way, but I think that there's some promising evidence that there's something to that. So next time you flip over a mushroom, you look at its gills. If its gills are super tightly packed, maybe that tells you something about its spores and how those spores are behaving in the air. Whereas if those gills are really spread apart, maybe those spores are being launched farther off of the gill surface. We got time for more questions here.
I think there's a comment, and the name is Copper, talking about mushrooms as omnivores. And actually, I think there is, unrelated to this too, isn't there a mushroom species that also attracts tiny bugs to eat them? But yeah,
Yeah, so I think what you're referring to, Kathy, are fungi. I mean, of course, there are the cordyceps fungi of The Last of Us fame that will, utterly destroy things like ant colonies. I've seen it with my own eyes. You find one of these ants with a mushroom growing out of its head, and then you find like a million of them, and it's like a horror movie. So I see why they did that for the video game and the TV show. But I think what is cool about yes, I was gonna bring up the oysters. So something cool that my lab mate Aishwarya Veerbahu has been looking into is invasive golden oyster mushrooms, and essentially trying to understand what kind of comparative advantage, if you want to describe it that way, like essentially what competitive edge they have over local communities of fungi that are evolved for, Wisconsin, Minnesota. Why are these local communities of fungi being pushed out by these invasive, non-native fungi? And one theory that may or may not be the case is that these golden oysters are really good at lassoing nematodes, right? And that might be a way that they are just able to get a lot more nutrition or energy or something from the environment that enables them to be so dominant in our forests. And I mean, if you haven't seen this, you're gonna see it. Just give it a couple weeks to a couple months. They're coming. Walk around the woods and you see some yellow mushrooms growing. Those are invasive golden oysters.
awesome. Yeah, but even talking about the ectomycorrhizal versus the saprotrophs, we were talking right before everyone joined here about the lecture that Christian Schwartz did about sort of like something that is symbiotic and that maybe it's not symbiotic in every instance, and that there are levels of symbiosis, and sometimes it might be acting more like a parasite, and sometimes it might be giving back more depending on the conditions and how it, like you were saying, there's this kind of like sliding scale of time and space and where it's being measured. Like when a plant is young versus when a plant is old and maybe is healthier and has more to give back, versus when it's young, the ectomycorrhizal fungi might be taking more than it's giving, and then gets to a place where it's able to give more. So I think some of those, yeah, it's fun to hear how everything's related, even though you started talking about spore shape.
Yeah. Yeah. I mean, I think that spore shape is really cool because it helps us simplify these complicated systems. And that is such like an appealing thing for us humans. You know, this is why we keep getting into trouble. So we want to fit these fungi into neat categories, like looking at a mushroom and say, this is an ectomycorrhizal mushroom. It associates with trees. It's a good mushroom. This is a saprotroph. It eats things in the soil. It's free living. This is a pathogen. It hurts these plants that we like. It's a bad thing. But in ecology, you know, everything is neutral. It's value neutral. It's simply self-interested. And if it's, you know, if it's evolutionary selective pressures make it such that it behaves well in partnership, then it will over time become a better and better partner. Or maybe the evolutionary pressure is basically just be an opportunist, you know. When there's good partners around, be a good partner. When shit hits a fan, you know, all bets are off, something like that. But spore shape is cool because it's well, it's not a single cell, but it's often a single cell or a collection of cells. It's a bottleneck. It helps us reduce this complexity to an essential part of the fungal life cycle. Every fungus you see in the world around you was a spore at some point in its life, and we don't understand what's going on with those spores. So I think that like studying these spores is a great way to understand something about the fungi that they turn into later on, and in a way that enables us to like simplify a lot of these like messier parts. And of course, you know, this project, it says, you know, there's complex factors behind spore shape, even though spore shapes are simple. So
Awesome.
take it with a grain of salt.
Thank you. Okay, I have one more question, and I'm going to ask it of you, and then I'll ask Michael to unmute and answer the same question. And it's just tell us about what you're going to do next.
Michael, would you like to go first?
Sure. Thank you, Jason. Awesome presentation, by the way. That is such a nice niche area of mycology. I love it. I'm not very good at physics, but it was really cool. So the question was, what am I doing next? Like in my career? Yeah. Okay. I mean,
or for fun, yeah.
I'm going to Rutgers in September, and I'm really excited to work with Dr. Ning Zhang on more obscure oddball fungi. We recently talked about a um, a separate project idea from describing new species from the New Jersey Pine Barrens, and that's looking at fungi associated with carnivorous plants, which there are a lot of in the bogs of New Jersey. They may impact these plants quite a bit. There was actually one study on a fungus associated with the sticky stuff of sundew, and apparently this fungus may be helping the sundew digest prey. So they may impact these plants a lot more than we once thought, and I'm excited to do these interaction experiments with these plants being inoculated with different fungal symbionts and seeing what do they do? You know, what do they do to these plants? So yeah, that's what I think I'm gonna do next.
Awesome. Thank you. Cool and obscure. Amazing. Jason?
All right. I just defended my thesis at the University of Wisconsin-Madison. So I am now apparently a Master of Science. I am going to, in August, move to Hawaii, where I will do my PhD, also in fungi, studying invasive ectomycorrhizal fungi. Essentially, the question will be, what makes good fungi do bad things? um, and I'm excited to spend my next 4 or 5 years out there working with a really good lab, really good PI, really cool study system. And then after that, I don't know, maybe to be a teacher somewhere would be really cool. I love educating. I see in the chat somebody asked about the Sclerotia project. I'll quickly plug it here, but basically part of the reason I am going out from wonderful Midwest, I know Hawaii sounds so exotic and whatnot, but I actually really like this area and I'm sad to go. Part of it is just that my funding got pulled while I was doing my research here at the University of Wisconsin-Madison. And this was a common thing. A lot of people I know, graduate students, had troubles with this. And we started to get to thinking, I suppose you never really miss a thing until it's gone, but we started to get to think about like, how does mycology get made? Like how does the sausage get made? And the truth is, it's a lot of scientists going, begging and scraping to either the government or companies or private individuals. And sometimes, mycological societies like you guys do amazing work pitching in as well to support grad students like me. Thank you so much. But in an ideal world, none of that would be necessary because mycological science would be funded regardless of political whim, shall we say. So we started the Sclerotia project. Essentially, the idea was fungi make sclerotium. They are hard knots of hyphae that allow a fungus to survive winter or drought or some harsh environment, only to thrive once the storm is past. And we're trying to make those basically in the form of just collecting some money from mycologists and mycological enthusiasts today. We're going to seal it up for 100 years so that none of us are alive when it comes out. And then in 100 years from now, people are going to say, holy smokes, look at all this money that past mycologists have left for us to do really good, cool citizen science or fundamental science projects that otherwise might not get done because of whatever. So if you go to the link, thank you, Kathy, for plugging this. If you go to the link there, you can see the webpage for our last year. We ended up raising, I think, $15,000 all through small donations. And that, I don't remember the ballpark number off the top of my head, but that'll be like 7 million or maybe $10 million in 100 years, depending on your calculated interest rate. So, quite a big amount of money. Too bad we can't buy a mushroom yacht with it today, but someone will eventually. So give us a check out, and if you got some money to spare, consider donating. We're gonna start it up for this next year shortly. And yeah, so thanks.
Amazing. Yeah, thank you. All right. Any last questions? Perfect. Well, this has been really great. It's, really fun. And I know there'll be other people watching the video. We'll share it with our membership